So in this video, we're going to look at the 3 stages of cellular respiration. And we'll be using this large schematic in front of you here. The teal part here is going to be glycolysis, and it's going to occur outside the mitochondria. This large kind of kidney bean shape is supposed to be a mitochondria. So the first stage of cellular respiration is called glycolysis, and it occurs in the cytosol. And in glycolysis, 1 glucose, 2 pyruvates, which are 3 carbon molecules, it makes 2 ATP and 2 NADH. This is a 9-step process, so it's been abbreviated over here. We start with a 6-carbon glucose. We make 2 ATP and 2 NADHs, and we end up with 2 pyruvate molecules, each having 3 carbons. So we haven't lost any carbons yet. This is the most widespread metabolic pathway, and so it's thought that it evolved very early because it is shared by almost every living creature that I can think of. Okay, some organisms, namely yeast and bacteria, stop their metabolism here. They make the 2 ATPs and then they're done. So let's go and fill this in on our big chart at the top. So I filled in the glycolysis. That's the name of the reactions. Glucose goes to make 2 pyruvates. 2 ATP are made down here, and also 2 NADHs are made. We'll be talking more about the NADH. For now, let's just say that it is a high energy molecule. And it has 2 electrons on it here and here that are going to be very important in the electron transport chain. So now we've made pyruvate. What happens to it? The pyruvate is then oxidized. First, it's transported into the mitochondria. So the pyruvate oxidation is going to occur in the mitochondria. This is our little diagram of it. Here's our pyruvate. It's got 3 carbons. We're going to lose a carbon dioxide in this reaction, and we're going to make another one of those NADs that I was telling you about with the high energy electrons. And we make an acetyl-CoA. It's a 2-carbon molecule. So just a little comment about our accounting here. First, when I put the 2 carbons here, that means it's a 2-carbon compound, just so we can keep track of how many carbons we've got. We start with 6, then we have 2 molecules of 3, and now we've lost a CO2, so we've got acetyl-CoA, which is a 2-carbon fragment. Note that we made 2 of the pyruvates, and so now we're making 2 acetyl-CoAs and 2 NADHs and 2 carbon dioxides. That's just because we have 2 of these pyruvates to go through this cycle. Now let's move to the top and fill in. So pyruvate oxidation goes in the orange square. And we want to note that we make carbon dioxide. That's coming down the gray circle below. And we've made another 2 of those high energy NADHs. And we'll see what happens to them later. The end result of pyruvate oxidation are we've made 2 acetyl-CoAs. And they're going to join the citric acid cycle, also known as the Krebs cycle. I may slip occasionally and call it the Krebs cycle, but citric acid cycle does just as well. And the citric acid cycle is indeed a cycle, as we'll see in just a minute. So the citric acid cycle occurs in the mitochondria. And it is cyclic. So what happens here is we have a 4-carbon compound that comes in and is joined with the 2-carbon acetyl-CoA to make a 6-carbon compound that starts the cycle. As it goes around a series of reactions, 2 carbon dioxides are given off, and that regenerates our original 4-carbon compound. In addition, we get 3 NADHs, we get an ATP, and a new high-energy molecule called FADH2. So for each glucose, we'll make 2 acetyl-CoA molecules. As it goes around, 2 revolutions of the cell cycle of the citric acid cycle, it'll have to go around twice because we've got 2 of them. We'll get 2 ATP, 6 NADHs, 2 of the FADH2s, and 4 carbon dioxides. So all 6 molecules of carbon have all been taken care of. All right. So this is our recap so far. We've made 4 ATPs. We've made 10 NADHs and 2 FADH2s and 2 and I don't have it down here, but on my key, but it's 6 CO2s. Okay. So the cell can use the ATP immediately. I've already told you the cell uses 10 million in an active muscle cell per second. But to use the high energy molecules of NADH and FADH2, we're going to have to use an additional step, an electron transport chain, to make ATP. Let's go and put the citric acid cycle on our big diagram. So here's our big diagram that should be probably at the top of the page, I think. We've put in the citric acid cycle in this orange circle. We've made 2 ATP. We've made 6 additional NADHs and 2 additional FADH2s. So far we've made our 4 ATPs, and we've made a bunch of these high energy molecules. We've made the NADHs and we've made FADH2. Those are going to feed into this purple box, which is going to be oxidative phosphorylation. So what you see here is an enlargement of a mitochondria. If you remember when we looked at mitochondria, the mitochondria has an outer membrane that I've drawn here in green and an inner membrane that I've drawn in blue. The inner membrane, as you recall, is all folded in on itself. This divides the mitochondria into 2 different spaces. So we have, let me get my pen going, we've got a space between the green outer membrane and the blue inner membrane that we're going to call the intermembrane space. That just means between membranes. And I'm not going to be able to, I ran out of room. But that's the intermembrane space. Inside the blue, we just have the matrix. Yes, and this is not the one where they wear the big black coats. Okay, so let's look and see what's happening here. This top membrane is the green membrane. This is the outer mitochondrial membrane, the one I drew in green. And this closer to the bottom of the page is the blue membrane. This is the inner membrane, okay? And you'll see that the inner mitochondrial membrane has lots of proteins that span the membrane. It's got protein 1, and then there's a protein 2, and there's a Q, there's a protein 3 here, 4 is here, and then we've got this large ATP synthesis. We've got lots of proteins that are spanning the membrane. We've got our high energy NADHs that are coming here, and they're going to offload their high energy electrons. And this is a high energy compound, the NADH. The NAD with a plus is a low energy compound. And the difference is it's offloaded its 2 electrons to this protein complex 1. Okay, when it does that, a hydrogen is pumped across this membrane. So we get a proton, a hydrogen, that's pumped in. But we're not done. Protein complex 1 will transfer the electrons to this Q, which will then transfer the electrons to protein complex 2. And another hydrogen ion is pumped into the intermembrane space. But we're still not done. The electrons are transferred to an even lower state to cytochrome C, and then finally to protein complex 4, and yet another proton is delivered into the intermembrane space. The electrons are then very low energy electrons, and they are combined with oxygen and hydrogen and make water. So we start with high energy electrons over with the NAD, and as we go through these complexes, we go to lower and lower and lower energy until finally we just end up with water. And as we step down, we pump protons into the intermembrane space. Well, now what does that do for us? Now we've got an excess of protons in here. We've got a whole bunch of them. And we've set up a chemical gradient. Remember when we talked about diffusion? These protons will diffuse out. They'll want to diffuse out, and as they do, they go through the ATP synthesis, and as they leave, they power this process where ADP, that's adenosine diphosphate, has a phosphate added to it and becomes ATP, our chemical that we use for all our energy transactions. So that's very complicated. There's a lot going on here. But the important things are that we start with a high energy molecule. We end with low energy as it goes in small steps down the energy gradient. And we make a chemical gradient that can then power the ATP synthase. So here's our summary. Electrons from NADH and FADH2. I didn't mention it before, but FADH2 has a little less energy, and it enters here at Q instead of entering at protein complex 1. So only 2 protons were transferred for it. But it's a very similar process. Okay, so the electrons from these compounds are passed down the electron transport chain, and they eventually end up on oxygen, which picks up protons to form water. As the electrons are passed to lower and lower energy states, hydrogen ions are pumped into the intermembranous space. Then, when the hydrogen ions diffuse out of the intermembranous space, ATP is made by ATP synthase. Oxidative phosphorylation will make about 32 ATPs. I'm sorry, 28. I did the total. ATP. So just oxidative phosphorylation makes 28 ATPs per glucose. So this number will vary because different cells have different efficiencies. So the oxidative phosphorylation makes a lot more ATP than the rest of the processes we've been looking at. But what happens in the absence of oxygen? So if we don't have oxygen, then we can't pass these electrons to anything, and we get blocked right here. And if you can't get rid of these electrons that are here, we won't be able to add any more here. So the whole process gets roadblocked and will not occur. So if you don't have oxygen, you can't have oxidative phosphorylation. So let's go to the top and add this to our overall summary. So this is oxidative phosphorylation. This is where we're going to use our NADs and our FADH2s. This is the step that uses oxygen and makes water. And we're going to make about 28 ATPs for a total of 32 ATPs. So in our final summary, we've got one glucose making about 32 ATPs and heat. Remember, that's why you're able to stay warm, is all that inefficiency. So the efficiency here is about 34%. Your car is only about 25% efficient. So the process is pretty good, except in the times that the body actually seeks to not be efficient. So in brown fat, the membranes are breached here. This membrane will have holes in it so that the hydrogen ions can leave without going through the ATP synthase. So the whole cycle will occur and heat will be generated, but no gradient will be produced, and so no energy will be supplied. And this is found, brown fat is found in babies, and it can also be found as a response to cold in adults. So next up is